I think we should go do Mr. Richardson's bio project.
Alright.
Let's go do it now.
Guys, where are we going?
We're going to Chris's mouth.
Oh, where are we?
We're in Chris's mouth.
What is a mouth?
The mouth is the first portion of the alimentary canal that receives food and is the location of starch digestion.
Look at those white blocks. They are teeth, the hardest substance in the human body. They help us to chew and break down food.
Oh, what is this thing beneath us?
It's the tongue, a muscular organ in the mouth of most vertebrates that manipulates food for mastication and is used in the act of swallowing.
What are these sticky things around us?
Oh, I know it. These are salivary juice. Salivary juice is composed of 99.5% water plus electrolytes, mucus, white blood cells, epithelial cells, enzymes, antimicrobial agents such as secretory IgA and lysosome. It has a pH level of 6.2 to 7.6 and it promotes the digestive action of moistening food, helping to create a food bolus containing enzyme, amylase, and break some starch down into maltose and dextrin.
Do you know where salivary juice comes from? They are secreted by salivary glands, which are exocrine glands that produce saliva through a system of ducts.
I've learned that saliva contains some enzymes named salivary amylase in it. These enzymes promote the following digestive reaction. Starch plus water equals maltose. Water is a crucial component of the digestive juice. In our mouth, saliva is water-based. In our gallbladder, water gives bile mucosal fluidity.
Ah! What's going on?
I think Chris is trying to swallow us.
Swallowing is an involuntary reflex triggered when the bolus is pushed upward by the tongue against the soft palate and backwards the pharynx. We are now entering pharynx, which is a region that receives air from the nasal cavity and food from the mouth.
Hey look, that's the epiglottis. It is a flap of tissue that covers the glottis and the throat that prevents food from entering the white windpipe and the lungs when swallowing.
We are now passing the esophagus, an organ in vertebrates through which food passes, aided by peristaltic contractions from the pharynx to the stomach. The function is to open and receive the bolus when swallowing occurs.
Peristalsis is a rhythmic muscular contraction that pushes the food along the digestive tract. Peristalsis begins in the esophagus and continues in all the organs of the digestive tract.
Look, that is the cardiac sphincter. That is the sphincter between the esophagus and the stomach. Relaxation of sphincter allows bolus to enter stomach, and contraction prevents stomach chyme from re-entering the esophagus.
We are now entering the stomach. The stomach is a bean-shaped sac located behind the lower ribs. Once food hits the stomach, sphincters at the opening of the stomach and the exit into the small intestine close. The lining of the stomach then secretes hydrochloric acids and enzymes that break down the food so that it can continue on its journey through the digestive system.
What are all these liquids?
They are gastric juice containing water, electrolytes, hydrochloric acid, enzymes, mucus, and intrinsic factor. Has pH value of 1 to 3. They can activate pepsinogen into enzyme pepsin and helps digestion by breaking the bonds linking amino acids.
Wait, if it is that acidic, why isn't the stomach melting down?
Because the mucus in gastric juice is protecting the stomach from the hydrochloric acid. Also, the mucus kills bacteria and gives fluidity to peristalsis in the digestive tract.
Hydrochloric acid causes the stomach to have a pH level of 2 or 3. HCl in stomach kills most bacteria present in food, but does not digest it. It also activates the digestive enzymes by changing the pH level from slightly acidic to very acidic. because acidic pH of 2 to 3 is optimal for the enzyme pepsin to digest protein in.
Curious about the digestive reaction? Let me teach you. Proteases, including pepsinogen, pepsin, trypsin, inside the stomach breaks down protein with H2O to create peptides. Let's get out of here before we hurt ourselves from the acid.
Let's go through that pathway.
That is the pyloric sphincter, which is a band of smooth muscle at the junction between the pylorus of the stomach and the duodenum of the small intestine. It plays an important role in the digestion, where it acts as a valve to control the flow of partially digested food from the stomach to the small intestine.
As soon as we enter the small intestine, we are now in an area called the duodenum. This tissue receives partially digested food, known as chyme, from the stomach and plays a vital role in the chemical digestion of chyme in preparation for absorption in the small intestine. Any chemical secretions from the liver, gallbladder, and pancreas mix with the chyme in the duodenum to facilitate chemical digestion.
That dark red, leaf-like structure must be the liver. The liver's main job is to filter the blood coming from the digestive tract before passing it to the rest of the body. The liver also detoxifies chemicals and metabolizes drugs. As it does so, the liver secretes bile that ends up in the intestines.
Then that organ located under the liver must be gallbladder. The gallbladder stores bile, a combination of fluids, fat, and cholesterol. Bile helps break down fat from food in your intestine. The gallbladder delivers bile into the small intestine. Bile salts and bile are derived from cholesterol, and they emulsify fat in the small intestine. When fat is emulsified, it breaks up into droplets, providing a much larger surface area that can be acted upon by pancreatic lipase, which helps to digest fats.
How about pancreas? I heard that pancreas secretes insulin for our body.
Yes, you're right. Pancreas produces insulin and other important enzymes and hormones that help break down foods. Insulin is produced from the beta cell in the pancreas in response to rising glucose in the bloodstream. Normally, the blood glucose level increases after eating a meal. When the level rises, the pancreas releases insulin in order to absorb glucose from the blood and lowering the blood glucose level.
The pancreas has an endocrine function because it releases juice directly into the bloodstream, such as insulin. It also has an exocrine function as it releases juice into ducts. The pancreatic juice is a good example. The pancreatic juice is composed of enzymes such as trypsinogen, pancreatic lipase, nucleases, and amylase. It has a pH level of 8 and it promotes digestive action of neutralizing the low pH of a coming from the stomach, assist in the breakdown of the protein, lipids, and carbohydrates for further processing and absorption in the intestine.
As pancreatic juice containing sodium bicarbonate is released into the duodenum, it lowers the acidity from a 2 or 3 to an 8 or 9. The addition of sodium bicarbonate to the acidic chyme makes the pH change from acidic to basic, which is essential because the basic pH of 8 to 9 is best for enzymes to digest in. The enzymes in our pancreas are pancreatic amylase and nuclease. They promote the following digestive reactions. Starch plus H2O becomes maltose. Lipid droplets plus H2O becomes glycerol plus fatty acids. Nucleic acids plus H2O becomes nucleotides.
Phew, that's pretty hardcore chemical digestion.
Now we have more. We are entering the actual small intestine. The small intestine is the part where 90% of the digestion and absorption of food occurs. The main function of the small intestine is the absorption of nutrients and minerals from food.
Look at these mucus around us. This is the intestinal juice, which also facilitates absorption as it combines with pancreatic juice to provide liquid medium. The intestinal juice composed of hormones, digestive enzyme, mucus, neutralizing substances released from the glands and mucous membrane lining of the small and large intestine. It has a pH level of 7 to 8.5, and it promotes the digestive action of neutralizing hydrochloric acid coming from the stomach, releases gastrointestinal hormones into the bloodstream, contains digestive enzymes that facilitates the digestion.
Which type of digestive enzymes can we find in the intestinal juice?
We can find peptidase in small intestine, which breaks down peptides with H2O to create amino acid, maltase in small intestine that breaks down maltose with H2O to create glucose, and nucleus in pancreas to break down nucleic acid with H2O to create nucleotides. The wall of the small intestine has folds that bear finger-like projection called villi, which give the intestinal wall a soft, velvety appearance. A villus has an outer layer of columnar epithelial cells, and each of these cells has thousands of microscopic extensions called microvilli.
We are at the end of the small intestine. Where are we heading towards?
We are going to enter the large intestine. The major function of the large intestine is to absorb water from the remaining indigestible food matter and transmit the useless waste mineral from the body. There is no digestive enzymes in the large intestine. However, we have some anaerobic bacteria. These bacteria break down some indigestible material and produce some vitamins that our bodies can absorb and use. They can also digest certain carbohydrates that humans can't.
Look at that finger-like tube. Is that the appendix? I got appendicitis before and it hurts.
True, the function of the appendix is currently unknown. One theory is that the appendix acts as a storehouse for good bacteria, rebooting the digestive system after diarrheal illness. Other experts believe the appendix is just a useless remnant from our evolutionary past.
Where are we heading towards? When does our trip end? I need to go home.
We are heading towards Chris' rectum.
What is the rectum?
The rectum is the lower part of the large intestine that connects to the sigmoid colon. It receives waste, stool or feces from the colon and stores it until it passes out of the body through the anus.
We're almost at the end of our trip.
Wow, having different pH level in various regions of my digestive tract really helps me digest well. If the enzyme doesn't reach the optimal pH or temperature, enzyme productivity decreases. Without optimal pH, we wouldn't able to digest.